Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Utilizing Built-in Antimicrobial Technology to Support Surface Hygiene and Cleanliness in Food Processing Environments. My name is Sydney, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Microban, who developed the content for this presentation. Part of Bar Brands International, Microban International is home to the most trusted and well-known global brands in the antimicrobial, odor control, and sanitization and disinfection markets, Microban and Ultrafresh. Its organization has experienced over 100 collective years of growth and has revolutionized the industry. As the global leader, its proactive systems keep products cleaner and control odors better by preventing microbial problems before they start. Microban International drives innovation by combining science and creative solutions that enhance high-quality consumer, textile, industrial, food service, and medical products. Today, the Microban and Ultrafresh brands and technologies are featured on thousands of products worldwide, including food storage containers, restaurant menus, dishwashers, cutting boards, and more. The company is headquartered in North Carolina, USA, with multiple operations in North America, Europe, and the Asia Pacific. And now I'd like to introduce our speakers for today's event. Dr. Glenna Richards is the Director of Microbiology and Analytical Chemistry at Microban International and has been with the organization since 2014. The focus of her current work is innovation and antimicrobial technologies, with an emphasis on assessment of technology and product performance and efficacy. She has worked in various capacities as a scientist, researcher, standards officer, teacher, and professor. Her experience spans working in government and regulatory agencies, academia, and private research organizations, and she's a member of various food industry organizations. Dr. Richards earns her Bachelor of Science in Biology and Master of Philosophy in Botany degrees from the University of the West Indies in Kingston, Jamaica. She earned her PhD in Food Science and Technology with an emphasis in food safety and microbiology from the University of Georgia, USA. Tara Conley is the International Director of Regulatory Affairs at Microban International. Leveraging more than 15 years of expertise in chemical regulations, Tara oversees regulatory compliance for international antimicrobial applications. This includes delivery of claims guidance, strategic regulatory advice, and on-demand compliance for support for Microban's antimicrobial business unit. Prior to joining Microban in 2019, Tara worked as a regulatory expert for chemical distribution, food ingredients, and household consumer products organizations. Tara holds an honor Bachelor of Science in Chemistry from the University of Toronto. And now, without further ado, I'd like to pass the presentation over to our first speaker. Glenna, you may begin when ready.
Good morning, good afternoon, and good evening all. Thank you very much for joining Tara and myself for this session, which is part of Microban's webinar series about incorporated antimicrobial technologies that are used to support various industries in which critical surfaces are susceptible to microbial degradation. Today, we will be addressing some hygiene challenges that occur in the food processing environment. Also, we'll be looking at interventions that are designed to address microbial risks in the food processing environment. We'll also look at the standard test methods that are used to objectively evaluate the performance of antimicrobial technologies. Then Tara will discuss registrations and regulations that govern the use of antimicrobial technologies in the food processing environment. And then finally, I will present results from a study done in the Microban facility where a technology that is registered for food contact and non-food contact surfaces was evaluated. Let's talk about the food processing environment. It is certainly not an overstatement to say that the food processing is a very complex operation. The food processor is required to confront and address issues related to sourcing of high quality raw materials and ingredients, quality control, record keeping, monitoring and traceability, packaging, and marketing. However, the requirements and demands of sanitary equipment design, good manufacturing practices, and sanitation interventions cannot be underestimated. Food processing is identified as a hygiene critical activity for three reasons. First, surfaces in the food processing environment are continuously exposed to microbes, such as bacteria and fungi, that are entering from various sources. Secondly, bacteria and fungi do not just die off, instead, they persist, especially if there are environmental conditions that support their ability to attach to surfaces and their subsequent survival and growth. These conditions include those that commonly exist in the food processing environment, namely high nutrient levels, uh, available moisture, and temperatures that are optimal for various microbial activities. Thirdly, the effect of an even small lapse in hygiene practice can be multiplied rapidly and give rise to large problems. Sources of bacterial and fungal contamination include air, water, raw materials, and equipment, pests, and personnel. If you think about it, raw materials that are entering the food processing facility are coming from various sources, and believe it or not, they are not sterile. Some foods are rich in nutrients, including sugars, fats, and proteins. Some foods contain high concentrations of water, plus the environment also has high levels of moisture from processing and cleaning activities. And there's also water found in drains, ductworks, and pipes. Environmental and surface temperatures may be in the optimal range for microbial metabolism. Let's not forget people. There are people involved in the food processing activities, and there are high-touch surfaces lending themselves to rapid distribution or spread of organisms that are introduced. Microbial introduction into the food processing environment presents challenges that are exacerbated if there are no or inadequate interventions, or worse yet, if there are hygiene failures. We can all agree that microbial activities on food contact and non-food contact surfaces present risks, including food product safety in the form of foodborne pathogens, food product quality by way of spoilage microbes that impact shelf life and brand reputation, physical infrastructure attacked by microbes that damage, deface, and destroy structures. When structural deterioration occurs, structures can crack and parts can fall off into food products by way of, and you would refer to this as introduction of extraneous matter into a food product. There are many different surfaces in the food processing environment which are potential reservoirs for microbes and their activities. Food contact surfaces typically are those that come in direct contact with food. They're required to be smooth, impervious, free of cracks and crevices. They should be non-porous, non-absorbent, non-contaminating, non-reactive, corrosive resistant, durable, and cleanable. These food contact surfaces include utensils, conveyors, choppers, cutters, hoppers, silos, bins, refrigerators. Also, surfaces onto which food may drip, drain, or splash are considered food contact surfaces. Non-food contact surfaces are surfaces throughout the food processing facility that do not come in direct contact with exposed food. Those are required to be designed and constructed and maintained to be smooth, non-absorbent, and easily cleanable. They include walls, floors, doors, pipes, drains, lighting, air and water systems, control panels, workstations, stackers, buckets, elevators, reclaimers, forklifts, restroom facilities. So anything you can think of in the food processing environment needs to be considered and protected. If surfaces are not hygienically maintained, they can introduce problems with respect to microbial persistence, multiplication, and then exacerbated by physical spread. From a sanitary design perspective, all surfaces should be subjected to a cleanliness standard of care and maintained in a physical condition that contribute to hygiene in the food processing environment. When there are failures in hygiene practices, there are financial costs incurred. When microbes are thriving on surfaces, they can excrete corrosive chemicals that degrade the surface and resulting in cracks and defacements. They can produce pigments that can stain surfaces, and they can generate off gases that cause odors. If a food processor is ambivalent about a product's quality, they will need to expend time and resources and to conduct investigations and possibly discard lots of potentially tainted products. These metabolic products can impact food quality and safety and possibly be responsible for compliance failures, investigations, and product recalls. For example, if an inadequately cleaned surface is sampled during a monitoring exercise and is found to have microbial populations above a critical limit, a recall of in progress or in process and finished products may be triggered. In a January 2021 Food Magazine survey in the United States, the results demonstrated that 51% of food processors were planning to increase their food safety budget. This tells me that most food processors agree that an ounce of prevention is better than a pound of cure, meaning it's easier to stop something from happening in the first place than to repair the damage after it has happened. Now it's time for our audience to participate, and we have a poll question for you.
All right, thank you very much, Glenner. So the question we have is, which of the following surface groups would you say is most important when it comes to having a built-in antimicrobial feature? So I will give our audience a few seconds to get in their responses. And what those include air systems, which includes filters, ductwork, and insulation, plumbing, sinks, faucets, drains, door frames and handles, tiles, countertops, floors, and walls, and storage, bins, containers, and boxes. So the question again, which of the following surface groups would you say is most important when it comes to having built-in antimicrobial feature? Air systems, plumbing, door frames and handles, tiles, or storage? All right, I see a majority of our audience has voted, so I'm going to close the poll and display those results. All right, so we have 30% for air systems, 7% for plumbing, 13% for door frames and handles, 40% for tiles, and 10% for storage. Back to you, Glenner.
Thank you, Sydney. Now let us look at the valuable interventions that can be used to mitigate microbial effects in the food processing environment. Because food processing is such a highly regulated industry, food processors are required to demonstrate that their hygiene intervention steps to protect products and consumers are safe and effective. These interventions are used to limit introduction of microbes into the processing facility, minimize cross-contamination between surfaces and transmission throughout the facility, reduce survival and growth of contaminants on food contact and non-food contact surfaces, as well as reduce the time between contamination events and microbial activity to adhere and multiply. Time is a very, very critical feature of microbial ability to adhere and to multiply and to have an impact on the surfaces. Food processors are highly invested in keeping the food processing environment safe. Government regulators are also highly invested. For example, in a report in the US government, annually, the federal and state governments conduct more than 1 million facility inspection, thousands of analyses in the laboratories, and unfortunately, they have quarantined millions of dollars worth of food products when there are lapses in the food processing environment. So we can see that this is a very costly exercise for both the food processor and for regulatory bodies. Conventionally, hygiene interventions include environmental control, workflow optimization, hygiene and sanitization. Environmental control includes hygienic design and construction of equipment and surfaces from appropriate materials that allow easy cleaning and maintenance. Also, placement of equipment to ensure that they're easily cleaned and maintained. Workflow optimization refers to separation of food production areas from non-food production areas. They also include separation of activities into different zones based upon the hygienic needs. Management of product and airflow throughout the food processing facilities are also included in workflow optimization. Hygiene and sanitization activities include the regular cleaning and sanitization protocols, because when these are compromised, microbes will take advantage of residues and eventually compromise surface integrity, causing crevices and cracks. Cleaning refers to removal of soil particles from surfaces by mechanical, manual, or chemical methods. Sanitization, however, refers to treatment of a cleaned surface, so you clean before you sanitize, So it's treatment of a cleaned surface with chemicals or physical agent to destroy disease-causing or spoilage organisms. A dirty surface is not clean. Microbes will become attached, metabolize available nutrients, survive, and grow, especially if the food processing environment has optimal conditions with respect to moisture and temperature. So we're talking about time, temperature, availability of moisture, and nutrition. These are the four rails that will support microbial adhesion, survival, and growth, and the effects that will come as a result of their activities. Microbes will produce waste products that are progressively and aggressively, that will and can progressively and aggressively degrade surfaces. And then we end up in a loop where the cracked surfaces are unhygienic because they create more cracks that trap and support further activities. Here is an example of a workflow optimization and environmental control working together. Here you can see the separation of the non-food production area from the food production areas. We also have separation of activities such as unpackaging and packaging from the actual addition of ingredients and preparation of ingredients. So you have the basic hygiene, medium hygiene, and high hygiene. And the food processor will have to be the one to determine which steps are the most critical and which are the most susceptible to contamination. Food products will flow from the basic hygiene to the high hygiene area, and then the air will flow counter to the food production or the food product flow from high hygiene zones into medium and then basic hygiene zones. So an example of your different interventions working and supporting each other. Now, Microban would like to introduce what we call the systems approach to hygiene in the food processing environment. Environmental controls, workflow optimization, and hygiene have been critical to operations of the food processing facilities. However, some challenges persist. Even the most well-managed food processing environment encounters significant challenges. There's continuous introduction of microbial contaminants into the food processing environment during normal operations. There are human lapses and instrument failures that will interrupt your optimizations. Chemical activity diminishes over time post-application. Cleaners, sanitizers, and disinfectants that are not residual, their antimicrobial activity will diminish over time. There are limitations to conditions under which some chemicals perform. For example, hypochlorite in your bleach over time will deteriorate, and in the presence of high levels of organic matter, you'll find the bleach is no longer having that killing action. There are limitations. So, for example, some surfaces are invisible and or inaccessible to extensive cleaning and sanitization activities. But that does not preclude or exempt that surface from contributing to microbial flora in the food processing environment. To add a layer of support to the previously mentioned interventions, the systems approach includes incorporation of registered and regulated built-in antimicrobials into food contact and non-food contact surfaces. These technologies do not wear off or wash away. They continue to work during the product's lifetime. For incorporated antimicrobial technologies to be used in the food processing industry, product registration by regulatory bodies is required. Antimicrobial product registrations require that the organization presents data that demonstrates safety, non-toxicity, and effectiveness of the incorporated product that are also called treated articles. One example is our silver technology. Silver has been used in various forms as an antimicrobial for centuries. It has been used on living tissues to treat burns, as a wound dressing, and neonatal eye drops. It has been used on inanimate surfaces, such as for sterilization of surgical instruments. The main advantage of the silver technology is the efficacy to control microbial populations at low concentrations. Studies have demonstrated efficacy at concentrations less than 1 part per million and minimal effects on human cells. On my screen here are two micrographs. The block on the left is a figure of E. coli in block A that is in the presence of, that is not treated with silver. And here, B, C, and D are the electron micrographs of bacterial cells treated with 0.2 parts per million of silver ion solution. On the right, in block A, is Staphylococcus aureus, again, untreated. And in blocks B, C, and D, you will observe that these cells are falling apart in the presence of the low level of silver ion solution. And this was published in 2008. At the same time, silver antimicrobial activities are what we call broad spectrum. It is clear from research that bacteria, yeasts, and molds are generally susceptible to the oligodynamic actions of silver ions, and assorted silver compounds. Toxicity to microbial cells is exhibited at very low concentrations. The D value, and D value is defined as the time taken to achieve a 90% reduction in viable count. And so the D value of silver can be seen here in this table. E. coli, Pseudomonas aeruginosa, Caenorhabditis albicans, Aspergillus niger. These are very different organisms, and you'll see that you can have 90% reduction in the microbial population within minutes. Silver ions do not have a single mode of action. The ionic silver impact a wide range of molecular processes within microorganisms, resulting in effects ranging from inhibition of growth and loss of infectivity, and ultimately cell death. Silver has been shown to disrupt bacterial metabolism by its effect on their enzymes and proteins, as well as on DNA. They cause physical holes in the bacterial cells and eventually dehydration. And you can see here over time the microbial population. So here we are at about a little bit over an hour. You're seeing the differences between the control, where silver is not present, and the treatment where silver is present. Currently, silver-treated surfaces in the food processing environment include air filters, faucets, door frames, countertops, walls and floor tiles, sanitary tiles. So much of these are food contact and non-food contact surfaces. I'm sure everyone is interested in knowing, so how do you assess the efficacy of these built-in antimicrobial technologies such as silver? There are several industry standard test methods that are used to demonstrate and measure antibacterial and antifungal efficacy of these technologies. The test data generated are required and are used for food product product registration, the treated article registration, as well as quality assurance. The antibacterial test methods that we're talking about here are specific for non-absorbent, non-porous materials such as polymers, foams, plastics, coatings, and non-absorbent textiles. And then there's another test for ceramic and sanitary ware surfaces. And then there are antifungal efficacy test methods for polymers, plastics, and coatings. And these are industry-wide accepted test methods to assess the performance of these treated articles. The ISO 22196, the JISZ 2801, the ASTM E28180 are synonymous test methods. They're just managed by different test organizations, but they essentially do the same thing where they're testing for antibacterial activity. They use a gram-negative and a gram-positive bacteria. The incubation time and incubation temperatures are the same, and the standard is a minimum of 2 log reduction in bacteria over the contact time. And as you can see here, bacteria recovered from untreated polymer sample, these agar plates are heavily laden with a lot of bacteria compared to bacteria recovered from the treated polymer sample. Testing on ceramic is similar to what is done on polymers, and it's 18 to 24 hours of contact time at 37 degrees Celsius. And as you can see here, the inoculum is being applied to the articles, and on the left, you can see from the untreated bacteria recovered from untreated ceramic, and then in the middle and on the right, the lack of bacteria recovered from treated articles. The fungal tests are more qualitative, whereby the results are visual. It includes use of a cocktail of fungus molds, Aspergillus brasiliensis, Penicillium funiculosum, Chaetomium globosum, Trichoderma viridans, and Aurea basidium pullulans. And this test, the samples are incubated at 29 degrees Celsius for 28 days. And pursuant to that, there is scoring: 0 for no fungal growth on the sample, and it goes up to 4 where more than 60% of the sample is covered with fungus. As you can see here, the top, we have treated samples, untreated samples, and then below we have treated samples that would be scored at 0. At this point, I'll hand over to my colleague, Tara, who will discuss the regulatory considerations.
Thanks so much, Glenner. Hi, everyone, and thank you for your attention and engagement today. The regulatory landscape regarding food processing and antimicrobial use in that space is jurisdictional and complex. My objective here today is to present a few key considerations that impact the use of antimicrobial material preservatives for use in food contact applications and treated articles on a global scale. To start with, there are regulatory regimes regarding food safety that must be considered. In the US, these regulatory requirements fall under the FSMA, or the Food Safety Modernization Act. In the EU, they fall under the Hygiene of Foodstuffs regulations, and in Canada, they fall under the Safe Food for Canadians Act and its associated regulation. These regimes mandate standards and testing requirements for food products to ensure that food production is done in a safe way to preserve the integrity of the food that's being made. In terms of food contact substances, or substances that come into contact with food but are not actually food or food ingredients themselves, these are often regulated separately, usually under an associated Food and Drug Act or health branch of the regulatory agencies, with the objective of undertaking a more robust risk assessment, assuming a higher exposure profile due to the potential for ingestion of the substance. The US regulatory options can include the food contact notification schemes, as well as listing under 21 CFR, or the Code of Federal Regulations, for each of the food contact substances. The EU splits the food contact regulation into not just regulations for food contact substances in general, but also into more specific regulations regarding the use of plastic materials for food contact. In Canada, there isn't a specific food contact regulatory regime, but there is a requirement to ensure that food products are compliant with the Canadian Food and Drugs Act. There's also a voluntary letter of no objection, or a LONO, that can be issued by Health Canada upon request for substances that may come in contact with food. Canada also had a reference listing of accepted construction materials, packaging materials, and non-food chemical products, which was maintained by the CFIA, or the Canadian Food Inspection Agency, which would have captured materials used in, for instance, tile materials in food establishments. But that list has since been set for reference purposes only and is not considered an approved substances list. Antimicrobial material preservatives are often regulated as pesticidal or biocidal substances with separate regulatory requirements than food ingredients. As an example of some of the more robust regulatory platforms that manage and facilitate registration of antimicrobials, the US EPA's FIFRA, or Federal Insecticide, Fungicide, and Rodenticide Act, the EU's BPR, or Biocidal Products Regulations, and Canada's PCPA, or the Pest Control Products Act, and its associated regulations, all are tasked with the evaluation of antimicrobial material preservatives based on physical, chemical, toxicological, and efficacy criteria to ensure that they're being used at safe levels in application. These regulations cover not just antimicrobials, but also plant protection substances, veterinary pesticides, as well as industrial biocides. But with respect to the use of antimicrobials intended for use in food contact applications, there are a few specific things to look for if you're interested in incorporating an antimicrobial treatment into your food manufacturing equipment for protection of that equipment from bacterial growth. The US has the Food Contact Notification Scheme for antimicrobials. The EU has both the Article 95 Product Type 4 listing, which is the product type approved under the BPR explicitly for food and feed use, as well as the EFSA, or European Food Safety Authority, who can provide expert opinions for the approval of antimicrobial substances for food contact applications. And as I mentioned earlier, there's also the Canadian Voluntary Letter of No Objection, or LONO, that can be issued by Health Canada. These allowances and opinions can provide both manufacturers and downstream consumers with the added confidence that regulatory health agencies have looked at the potential risks associated with the use of these antimicrobials in food contact applications and have either given their approval or their no objection to their use. Once a surface is treated with an antimicrobial, it's considered a treated article, provided that the claims on that article are for the preservation of the treated item. Each jurisdiction mentioned has its own set of rules that govern the allowable claims and specific requirements pertaining to treated articles. Universally, though, there is no requirement for registration in any of these jurisdictions for treated articles that claim preservation of the article. Registration is only required if a cross-contamination or public health claim is being made. The claim sets are varied depending on the jurisdiction, so it's very important to properly understand the landscape before confirming any marketing materials for a treated article. Outside of those regulated regions, there are also international advertising laws that must be upheld in any advertising that's done on any treated goods to ensure that the claim sets are permitted in the region of sale. Microban has options for companies looking to add product preservation to their food processing equipment. And an example of this would be our SILVERSHIELD technology, which Dr. Richards will be speaking to in further detail in just a moment. But from the regulatory side, SILVERSHIELD technology is permitted for use in a variety of food-related applications per the EPA label, and has both the appropriate product type and EFSA opinion as referenced for the EU. There is also a safe history of use with this technology, not just in food contact applications, but in a variety of applications with high exposure potential similar to that of food contact. Lastly, we are living in unprecedented times, not only because of a global pandemic, but also because of environmental concerns, which are driving many companies to look for enhanced sustainability options for their businesses. The use of antimicrobials in built-in applications can certainly help companies achieve these goals indirectly, not only because of the reduced waste in equipment and contaminated food that can arise if components in the manufacturing process degrade or become contaminated with high bacterial loads, but also because that enhanced hygiene seen on the surface when antimicrobial technologies are used in these applications can also mean you spend less time and water, and therefore less money and man-hours, engaging in continuous cleaning programs to maintain the necessary level of hygiene. I'll shift back to Dr. Richards now for a more robust overview of our SILVERSHIELD technology. Thank you.
Thank you very much, Tara. We'll now take a look at the Microban SILVERSHIELD study that was conducted in our facility in 2015. Just a bit of background. Some food processing facilities require processing and storage to be done at refrigeration temperature of 4 degrees Celsius, which is about 40 degrees Fahrenheit. Listeria monocytogenes is a bacteria of concern in the food processing environment. A unique feature of Listeria is that compared to most other foodborne bacteria, is its ability to survive at such low temperatures. Not only to survive, but to multiply. In the United States, during the decade 2005 to 2015, Listeria was associated with 18% of all food recalls, representing more than 9 million pounds of food. That's a very expensive problem. In the United Kingdom, food industry cleaning tools and utensils have long been identified as a major source and vector of cross-contaminants. In 1998, United Kingdom government funded a study, and the data showed that non-food contact surfaces were significant sources of Listeria in the food processing environment. Out of 10,000 environmental samples, 17% of floor samples were attributed or were contaminated with Listeria. 25% of drain samples were positive for Listeria, and a whopping 47% of cleaning equipment were positive for Listeria. So in 2015, Microban conducted a study, and the objective was to examine the antibacterial efficacy of the Microban SILVERSHIELD study that is incorporated into powder-coated steel shelving units in refrigerators. And so the study was conducted at 4 degrees Celsius using the ISO 22196 protocol. The samples included acclimated powder-coated finish on steel coupons. They were inoculated with about 100,000 colony-forming units per milliliter of Listeria monocytogenes. And this Listeria that we use is a CDC strain, so it is a very robust strain. Dishes containing the inoculated coupons were incubated in the refrigerators at 4 degrees Celsius, and we assessed Listeria survival at the time of inoculation, so we knew how much we had put down at 0 hours, 2 hours, 4 hours, 24 hours, 48 hours, 72 hours, and 1 week post-inoculation. The data shows that Listeria can survive on untreated powder-coated steel refrigerated surfaces for an extended period of time. The Listeria population recovered from the coupons was reduced by 90% within 24 hours. So the difference between the untreated coupons and the treated coupons, you can see a greater than 90% reduction at 24 hours, greater than 99.9% within 48 hours. And 100 is the limit of 100 colony forming units per milliliter is the limit of detection. And so this is that's why you see that baseline. The comparative data gathered from these studies showed that the Microban SILVERSHIELD technology is effective in limiting the survival and growth of Listeria that was inoculated onto powder-coated steel refrigerated surfaces. This concludes our presentation, and more details on this study can be found on the Microban website, www.microban.com. And on behalf of Tara, I'd like to say thank you very much for your attention, and I will hand it back over to Sydney.
All right, thank you very much for that insightful presentation. Now I'd like to invite our audience to continue sending in their questions or comments right now using the questions window for the Q&A portion of the webinar. So I've already received a few questions and I will start with those. So our first question, do any of these treatments affect efficacy of phage usage?
I'll take that question. Bacteriophages are viruses that attack bacteria. These technologies can be used in parallel with other technologies that are being used in terms of hygiene. In order to determine whether or not the bacteriophages will be negatively impacted by the Microban technology, studies would need to be conducted before such a claim can be made positively or negatively, and we do not have that data.
All right, thank you, Glenna. Our second question, how much of the silver ions must be available on a surface to be effective in food facilities?
One thing that research has shown is that the efficacy of the technology increases with the amount of silver ion that is present on the surface. So as the available silver ions increase, the activity and the efficacy will increase. Before a product is registered or before you can make those claims, you would need to do studies to determine the concentration or the treatment level at which you can observe or you can see efficacy activity. So there's no one size fit all. It would need to be studied and evaluated for a particular environment or a particular application.
All right, thank you. Our next question. The ancient Egyptians used copper utensils for food hygiene. Have you looked at the use of copper in surfaces?
No, well, copper is not within our, that's another application, and that is not within our portfolio at this time.
All right, thank you. Our next question. How critical is it to have antimicrobial treatments on walls of food processing facilities?
Oh, very important. If you think about it, any surface that is open to contamination, once microbes are thriving, those microbes are going to cause staining, deterioration of your surfaces. I mentioned before that food processing facilities have high nutrient concentrations, high levels of moisture, temperature, there's constant introduction. And so surfaces that are exposed to splashes, draining, um, contamination should be protected because over time you're going to have that deterioration. And Tara mentioned the aspect of bringing it all together with reducing the time spent trying to mop up and to clean up, not saying that you don't need to do your hygiene activities, but this is a support for your routine cleaning and hygienic activities. And so anything you can do to maintain and retain infrastructure will be very valuable.
All right, thank you. Our next question. Does the silver leach out of surfaces?
No, silver is not a leaching technology.
All right, thank you for clarifying. Another question. Will your antimicrobial technology wear off over time, especially if it's applied as a surface coating?
Antimicrobial technologies that are incorporated into the structure of the material, as well as those that are applied in coatings, are intended to provide support for the conventional hygiene practices of cleaning, sanitization, and disinfection, and therefore they do not wear off. They continue to work for the use life of the product.
Wonderful. Our next question. Is there any location in a food processing environment that Microban antimicrobial technology should not be used?
Microban's portfolio includes various antimicrobial technologies, and some are registered for use in the food processing environment and some are not. If a technology is registered as being suitable for the food processing environment for food contact or non-food contact, um, once it's registered, it is it can be used anywhere in the food processing facility, whether it's seen or unseen, polymers, metals, ceramics, walls, floors, spigots, handles, low, medium, or high zones.
All right, thank you. Are all Microban antimicrobial technologies registered for use in the food processing industry or only some technologies?
I can take this one. Thanks so much for your question. As Dr. Richards mentioned, Microban certainly does have a broad arrangement of antimicrobial preservatives. Some of them are permitted for use in food contact applications, others are not. Microban is the global leader in antimicrobial technologies, and as such, we have a comprehensive suite of products to serve all sorts of built-in applications. So we're happy to work with customers directly to make sure that the right technology is being used to maximize performance, but also meet regulatory requirements.
Thank you, Tara. Our next question. What technology do you have for use in plumbing, so PVC pipes, with exposure to irrigation water with plant nutrients? Will there be concerns of leaching in water?
Well, silver has been used as a water treatment to make non-potable water potable. So I would not be concerned about having a silver treatment in a PVC pipe for water.
All right, thank you. Our next question, does Microban evaluate antiviral performance and efficacy of its technologies?
We do surrogate work with bacteriophages, and we do not do it internally in-house, but this is something that we do in collaboration with external third-party labs.
All right. If silver is not a leaching technology, how do silver ions kill the bacteria? Don't silver ions penetrate inside bacteria?
Yes, but when I think of leaching, my impression of the question was leaching out into environments far away from the actual source. It's acting on organisms that are on its surface and having that killing action. So my definition of leaching is not leaching away into the environment and having an effect. So the ions are actually on the surface, and the moisture on the surface creates the environment for the ions to enter the organism and have its killing action.
All right, thank you for clarifying. Our next question, what is the mechanism of kill if silver is bound and incorporated into the bulk of the material?
What is the killing action? Well, as I mentioned just in the previous response, the ions in the silver will will move from the incorporated surface or in the material, and they will move into the organism in the presence of moisture, and by chemistry will attack the organisms.
All right, thank you very much. I think we have one more question, and that is, from a regulatory perspective, can Microban silver antimicrobial technology be used for applications in Canada?
I will take that one. So that's a great question and a challenging question because the Canadian landscape is slightly complicated. There is an information note on the Health Canada website which speaks to specific treated articles requirements. I will say that for the use of antimicrobial manufacturing in Canada for silver, that is not currently permitted because silver itself is not registered in Canada, but there are some options available for customers who are interested in using silver technologies north of the border. But that is a conversation that we have to have with bespoke customers for specific tailored solutions. So I would invite that questioner to reach out to us directly and we can have an offsite conversation.
All right, thank you very much for those answers. We have reached the end of our Q&A portion of this webinar. But if we couldn't attend to your questions, the team at Microban may follow up with you. Or if you have any further questions, you can direct them to the email address on your screen. Thank you everyone for participating in today's webinar. You'll be receiving a follow-up email from X Talks with access to the recorded archive for this event. Now, a survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now, please join us in thanking our speakers, Glenna Richards and Tara Conley. We hope you found this webinar informative. Have a great day, everyone.